Thank you, John, for the invitation. It's a pleasure to be here with you all. There are some of you that there are a long time that I don't see, so I'm pretty glad here in the South America branch of the minimal cell. Well, so I'll talk a little bit about what we have been doing here in Embrapa, and we have been testing a lot of different genetic tools for the engineering and control of novel biological process at JCVI Syn3B. So one of the how can I go forward now? Okay, I have to go through here. Okay. So one of the tools that we are applying nowadays are the Syn cell strategy, which was developed for the Oxford group. Dr. Wang, he built an expression vector which expresses an endonuclease, the ICO1, which is capable of to degrade the chromosome DNA and the plasmid DNA from the original DNA from different microorganisms. He tested using 3 different microorganisms, including E. coli. And the idea here is to develop, a non-replicable functional synthetic cell. So once you degrade the chromosomal DNA of the microorganism, you have only an empty cell, and which with all the enzymatic soup that are present in the cytoplasm. But even though this cell without their DNA are still capable of doing specific functions according with the genetic construction that you insert inside of it. So our main idea is to use this strategy as a manner of developing new synthetic cells, and also to try to develop systems that to maximize the use of energy for a specific purpose. And at the same time, you can apply it to evaluate different function of metabolic pathways without interference of other genes present in the original bacteria. So we have tried to use this strategy using the JCVI Syn3B, the Syn1, and also Mycoplasma capricolum. And our strategy is we are trying to insert 3 different constructions. One is, of course, the endonuclease plasmid for chromosome destruction, which is...
Danny, can you repeat that? We lost you for a second.
Oh, I'm so sorry. Okay. So... just this slide, John?
Just like 10 seconds previous.
Okay. So,
sorry.
No, that's okay. I am sorry too. Okay, so just coming back. So this is the strategy that we are going to apply, trying to destroy the DNA from the synthetic cells, okay? And actually, the idea is to express the endonuclease ICO1 and also 2 other plasmids. The idea is to insert the adenine deaminase pathway as our system checkup. Let's see, once we have destroyed the DNA from the minimal cell, we want to check for how long the cytoplasmatic enzymes are still functional, and they're capable to produce the proteins necessary to move through all this metabolic pathway. And this is easy to check because the production of this pathway will produce oh my God, NIG3 and plus CO2, which will go down the pH of the medium, the culture medium. So it's easy to see. You can check only with the change of color using the pH of the medium. So it's easy. And another thing which is interesting is this pathway also produces an ATP. So it can also add more energy to the system, and maybe it will also be able to get to the cell a longer life, let's say, not life, but at least the system will be functional for longer with another ATP. And also the third construction would be a Q switch, because the system is functional, but even though we have some cells that are capable to escape of the endonuclease. So they have safety manners that are able to knock out the endonuclease which was inserted and just to start replicating again. So for these, we are going to apply a kill switch, which we will intend to use a CRISPR-I strategy to knock out essential gene and kill the remaining bacteria that survived to this strategy. So for these, we have tried just inserting the endonuclease by itself under the control of a tetracycline promoter. And we were able to see that the system in the first experiments have worked. So we have here the bacteria not induced. So here we have the cherry expressing normally. These are Syn3 expressing cherry, and so here we can see the bacteria, they are happy, they are growing, and they are expressing cherry perfectly. And here we can see with DAPI the chromosome side. So when you induce the system and the endonuclease is produced, you can see that the mostly of the cherry that was expressed is vanishing, and we can't see any DAPI anymore. So no DNA anymore in the cell. So these were analyzed after 48 hours of induction, and here you can see also the same thing, like with with tetracycline induction, the endonuclease stops the growth of the cells, and here it's the control with no tetracycline present, so have a normal growth, and then of course the cells start dying again. So our first analysis have shown that the endonuclease is functional in Syn3. And the second step is, okay, so let's see if we insert the ARC genes back, we will be able to check the function of this metabolic system. So just checking for the activity of the adenine deaminase pathway, we just observe the difference in pH of the medium, and here we have MCAP, which has the adenine deaminase pathway, the Syn1, which doesn't have this pathway, and here we have our clone, which is the Syn1 with ISO and the ARC genes included. So here, MCAP, you can see after 13 days, we can see there a difference by 1 of the pH, and here we have with the Syn and our clone, the Syn with the ARC genes also showing this difference, a difference of 1.5. It's considered a positive metabolic pathway activity. So Syn1 doesn't show any kind of changes. After 14 days, everything goes down, but we believe that here the cells are already dying, and so there's no difference here in the presence of the adenine. So, okay, we have both plasmids, the ICU and the ARC genes working. But within time and doing other analysis, we realized that our system wasn't perfect. The endonuclease was being deleted by bacteria, so it wasn't very stable. So we decided to introduce the integrase 9 as a control for the expression. So the integrase are capable to recognize specific sequence sites in the plasmid here in the construction. So where each turn it on and off the gene. So it's just changed the direction of the gene. So the original construction, you have the integrase under the construction under the control of the TET promoter, and our endonuclease in a different position in the different position for, against the promoter. So it's not expressed in this condition. Even with the so once we put the tetracycline in the system, it activates the integrase 9 expression, and then the integrase will turn on or just switch back the gene on the direction of the strain, and then the endonuclease starts to be produced. So with this, we acquire for sure a better system. And here, if you can see, using DAPI2, In this time, we have the Syn3B, which is our negative control with and without the tetracycline induction, and there is no difference between treatments. The same thing here we can see with the integrase 9 expressing cherry. Also, there is a slight difference between the curve growth, but not many. So maybe the integrase is kind of, we've thought that this might be giving a hard time for this to grow. So maybe we have some interference in here with the integrase. But when you see our treatments with the endonuclease, we see a much more different, higher difference between the cell growth, showing that the expression of the endonuclease is being activated and the endonuclease is degrading the DNA. But just to make sure that the integrase is not exactly also doing anything wrong or getting a hard time for the cell to grow, we also did some dilutions with our transformants and also with Syn3B. And so here it's after 24 hours of growth, we can see the integrase and cherry growing okay perfectly, as the same Syn3B is also growing. But we can see that our clones are not growing as the controls. So after 48 hours, the Syn3B actually most of them have grown, and also the integrase 9 within cherry, but our clones, the induced ones, are not growing as the negative controls. So, but even though we still see some scapes, we still see some cell growth. So now we needed to move for the kill switch. And just for you to have an idea, just to check, like just the plate also with tetracycline and without tetracycline controls, and you can see the colon is growing perfectly here, but not here, and also here, but not here. So, okay, checking. So we have our degradation system is working. So now we need the kill switch. For the kill switch, we have a strategy, which is to put everybody else under the control of the tetracycline promoter, but we would like to test different promoters and to be able to induce the expression in different time frames. So for that, we decided to try some inducible promoters that were developed for Mycoplasma pneumoniae, and we constructed them and inserted into Syn3B. We just did as one. These are all preliminary data. So we just did one induction, and after 24 hours, we were able to see that this construction here with 2 transcription factors and the promoter seems to be respond as we were expecting, although we still have a very low expression of cherry, fluorescence of cherry, differently when you see our positive control, which is the promoter and the tetracycline, which goes 10 times higher than we can observe here, even 100 higher actually. Here with this one is a little bit higher, but the difference between the treatments and even without the IPTG in this case, it's not too different. So we might need to do some work with these guys here, these promoters, and get them a better control of the expression. But when you see the original work in Mycoplasma pneumoniae, you can see that they have a higher fluorescence emission up after 50 hours to 60 hours. And here we just evaluated with 24 hours. So we will do a new evaluation to see if you can get the same results as for Mycoplasma pneumoniae. Although we would like to improve it and get the expression even faster than this. So maybe, I think it was Ben who was saying about the ribo switch, that for mycoplasmas, maybe we could try one of those transcriptions promoters to control these expressions. And well, and this is just to let you know that's another thing, another work that we are doing now. It's atomic force microscopy for the Syn1 and Syn3 to study not only surface topology, but also the viscoelasticity of the membrane. So this is just also, as I say, just we have, unfortunately, just have only preliminary data so far, but this is a very exciting field, and I think that it might help also in comparing the characteristics of the membrane and the genes that are being expressed in Syn1 in comparison with Syn3. So I would like to thank everybody. Thank you for listening. Thank for my team, Marco, Raquel, Mariana, Elívio. So everybody here worked hard, is working hard with the minimal cell. Thank you very much, guys.
Danny, you guys have made a huge amount of progress in this since the last time. Well, I mean, we talk about this regularly, but you're getting closer, but it's still a leakier system than one would like. I know we have several folks on board, like the group from Bordeaux, who have been developing some systems for genetic expression control. Do you have any suggestions for Danny based on what you've just seen? Sorry to put you on the spot, Pascal and Carol.
Yeah, hello everyone.
Hello.
Well, most of the tools that we have developed are not, to get, the fine tuning on gene expression. It's much more to get mutants. So, I'm not sure we have fantastic clues for this.
Yeah.
Or anyone else. I know we've got the folks from Canada and maybe a few people from Barcelona. Or not. But
you can send me emails, no problem.
and on Monday, Maria-Luc Sinar is going to talk about some of their things with pneumonia, so she may have some advice for you. I don't think she's on today, but Danny, this is a huge amount of progress, and I'm anxious to see how things develop with the atomic force microscopy.
Oh, me too. Me too.
We have thinking about membrane curvature. There are a lot of things that we're curious about.
Yes, teachers for the next workshop. Then you can present the whole picture. And I think that this can add some information too for the lipid composition and maybe different composition of the membrane, the lipids in the membrane. We could also try to characterize these membranes and see how different they are, if it can change the viscoelasticity and how they behave. This can also be an add-on on these sites. But thank you for listening. Thank you, guys.